biological sciences. So I hope you enjoy the talks today. Okay. I will give to the first host.
So the first speaker is going to be Michael Ng from the Hammer Lab, and he's been in the Hammer Lab for a long time now because he was a member of the lab as an undergraduate and now for his PhD. And he's got plenty of work to show, and he's going to be showing work that has been revised and resubmitted to MBio, so that should be published soon.
Hello. Does it work? Hello? Okay, it works. I guess I have to get used to it after 2 years or 3 years I've been using this. All right, thanks everyone for coming. My name is Michael. I'm from the Hammer Lab. Today my talk is going to be the type 6 secretion regulation in Vibrio cholera. So before I show you the bug, I want to show you a cute picture of something. No. No. Oh, there you go. Oh, yeah, it's my mouse. Well, how do I go back? There you go. All right, so before I show you the bug, I want to show you the cat. So I think some of the people over here has a cat. You know, they have a lot of different Cattitude personalities. You have a house cat that can be pretty quiet, pretty chill. And when you spray it with water, they can get mad and they get angry. You can have a feral cat that is always angry all the time, right? So that happens in bacteria as well. So in our lab, we study a bug called Vibrio cholera, and they can get aggressive. And you can find their house pet as our human isolate as a house pet. So human isolate get confusing because this doesn't work. Yeah. So you can human isolate that isolated from human, and they have a secretion system called type 6 secretion system, using it as our killing machine. So we call it as like an aggressive behavior, and it can use it to poke through the neighboring cell and kill the target cell. And in human string, it's known to be inducible, so it requires environmental signals, just like the water spray. It requires environmental signal to trigger the killing mechanism and kill the target cell. We also have environmental strings, just like the feral cats. They are always angry. Their type 6 secretion system is always on for some reason. It kills the neighboring cell without any known environmental signal. It just kills all the time. All right. So my project is trying to find out what caused the difference in the genetic level. So when I compare different strains, so within human string, there are 2 categories. We have HS1, that's a human string 1, that is CRISPR dependent. CRISPR is a transcription factor that affects the transcription of their type 6 genes. And then the environmental signals that turn on CRISPR, and CRISPR turn on type 6. So that is HS1. And HS2 has some internal signals that turn on T4 and turn on type 6. And this is a weird house pet that is always angry as well. All right? And then for the environmental string, we have our ES1 as a representative. We don't know what the signal is. We don't know what their transcription factor is, but their type 6 is always on. So these are the 3 categories. And this is the bug that I'm focusing at, trying to find out what the differences is between these 3. All right? So when I look at genomic level, so I start looking at the DNA sequence. This is pretty much all we know about the regulation of type 6. We have a type 6 gene here. We have a 5' UTR, but we don't know where the transcription start site is, so we don't know how long the mRNA is going to be. We have our CRISPR and T4 that we don't know where they're binding to, where they're regulating at. So when we zoom in this sequence, this is pretty much how it looks like. This is the HS1 sequence, and this is a cartoon from the previous slide. When I compare to ES1, you see a lot of SNPs. So that first give me an idea like these SNP probably contribute to the regulation. So maybe some of the SNP affect the transcription regulation, some of the SNP may be embedded in the mRNA that affect the post-transcription regulations, right? But there's a lot of SNPs, so I don't know where to start. So I put another string that always kill, which is HS2. So HS2 has only one SNP compared to HS1, which is the G to T mutation right here. So and the difference in type 6 between HS1 and HS2 is one is inducible and one is always killing, right? So this gives me an idea like maybe this is the SNP that is important in terms of like regulating the type 6 to make it inducible to always on. So but I still don't know where are those SNPs. I don't know where the mRNA starts. So I used a technique called 5' raise. It's not a frame shift. So I should have mentioned the ATG starts right here. Yep. So everything here is within the intergenic region that is not being translated into protein. So I did a 5' raise trying to find out where the transcription start site is. And I found that the +1 transcription start site is actually right here. So everything behind will be transcribed into mRNA. So it has a pretty large 5' UTR. And the promoter is right here. And then this is a -68 relative to the +1. All right. So this mutation is not going to be encoded in transcribed into mRNA. So it's going to, if there's any effect, it's going to affect the transcription of the type 6. And everything below is going to perhaps affecting the post-transcription regulation of the type 6, right? Because it's in the mRNA. So the first thing I try to do is to play around the intergenic region, replacing one with the other, and see how does it affect the type 6. So I did that in an HS1 background. So I did that in a killing assay by putting Vibrio, Cholerae, and E. coli together and see how they kill each other and count how many E. coli survive. So the higher the bar, the more E. coli survive. The less the bar, the less E. coli survive, right? So so the VASK is the type 6 minus control. So you see a lot of E. coli survive. CRISPR star is their activator of type 6, so it kills by fall log. Wild type doesn't kill because it doesn't have the environmental factors. When I replace the intergenic region with the HS2, remember there's only 1 point mutation from G to T, it starts killing again. And that's only 1 point mutation that triggers the type 6 killing. And then when I knock out T4, which is the regulator in HS2, it stops killing a little bit. It's not like the type 6 minus control, but it's decreasing. When I replace the intergenic region from the environmental string ES1, it kills really well. I got no survival. When I knock out T4, there's no survival. So just by replacing the intergenic region, I was able to change the type 6 activity from inducible to always on. And that's probably, and the most impressive thing is there's only one SNP between HS1 and HS2. So I did the opposite thing, reversing the mutation in ES1, which is the string that are always on. It has a T originally, and I replace it with a G. And it stops killing a little bit, not as much as the type 6 minus control, but it does stops killing by 2 log. And then when I knock out CRISPR, which is the regulator in HS1, it doesn't really affect the killing. When I overexpress the CRISPR, it kills again. So by making a T to G, I was able to convert type 6 to be an inducible killing mechanism. And that is CRISPR dependent, right? So yeah, next thing we wonder, is it is ES1 an exception? Is it like a conserved thing? So I look at a lot of different environmental string and human string in our lab that we sequenced like a couple years ago. We have a list of environmental string that all has a T in the -68 sites. And they have been shown to be always on. The type 6 is always on. with one exceptions. And then we have a list of human string that subset of them has a T that is always on, and then a subset of them is inducible that has a G. So this is pretty conserved to what we found, that T, when they carry a T, their type 6 is always on. When they carry a G, their type 6 becomes inducible. Bottom G. Oh, this one? Yeah, that's one of the exceptions I forgot to mention. Yeah, so there's two exceptions here. There's one exception right here that has a T that is inducible. There's a G right here that is always on. That's also another exception. We don't know what the reason is, but there's also other SNPs within the intergenic region. So those might play a role as well. And Sophia, the undergrad in our lab, tried to test a few additional environmental strings to see if they are also CRISPR independent and T4 independent. So the first figure I show you is the same ES1 that doesn't really, T4 affect a little bit, CRISPR doesn't really do much. And then when I show you ES2, ES3, and ES4, she found that they all have the same pattern. So wild type kills really well, T4 has different contribution in different strings, and CRISPR doesn't really do anything in these environmental strings. So we think in the environmental string, CRISPR doesn't really matter. It matters more in the clinic, in the human string. But T4 has different contribution in different strings. So we think T4 probably, well, the G to T may play a little bit role, but T4 may also act on other mutations somewhere else in the intergenic regions. So far, this is the old model that I showed you at the beginning. And within this few years, I was able to show, I was able to find out where the promoter is, where's the +1 is. So we know the mRNA starts right at the T here. And then I found there's a lot of mutations within the mRNA. So that probably contributed to the post-transcription regulation. I found there's a GT right here. Just happened to be GT. We found a GT mutation here that affect the type 6 regulation. Either they can have a G that is inducible, CRISPR dependent, or they can have a T that is always on and become CRISPR independent. Yep, that's what I'm trying to show. And we think T4 may have a little bit contribution as well. Like there's other SNPs right here, right? So maybe T4 is working direct or indirectly with these post-transcription regulations. And with that, I would like to thank my lab for the support and advice. I would like to thank Sophia especially, who helped me to do a lot of killing assay. Gabby, who, I don't know if she's here, but yeah, Gabby did some microscopy. I didn't show it right here. And my committee for tons of different advice. All right. Oh, any questions? Mm-hmm.
In cholera, so my question is, why do you see constitutive expression in environmental strains, but conditional in human? You would think maybe it would be important for infection, but I just don't know.
Yeah, so we don't know what the theory, we don't actually know what the reason is, but we have a theory that human string may want to damp down their aggression inside the human string when they are inside the human, so they are not triggering their immune system, whereas when they are outside of the human, they want to turn on their type 6 and prevent and help them for competing resources like space, nutrients, and prevent invaders.
In your survival bar plots, is that at multiple time points or a single time point?
It's only a single time point. We usually co-culture them for 3 hours and then count how many E. coli survive.
Do you expect those values to change if you had like doubled that amount of time?
We never tried that. We did that in microscopy just to see how what's going to happen. It's easier because it's automated. And we usually saw complete loss of E. coli, but not with this environmental strain. We only tested with the human strain and see what happened. Yep.
Have you tried those exception strains in your killing assays?
Exceptions?
The strains that don't, no?
Not yet.
Okay.
No. It's been tested before by Erin, our previous PhD student. She tested that, and she was able to induce the killing with chitin, which I didn't mention right here. She was able to induce type 6 with external signals. But no, I didn't retest that.
And if you go out, I mean, I really like the idea of looking at the intra-strain variation. I was going to ask about that, and you showed that. But if you go out further from that region, can you identify other potential mutations of interest by virtue of the relative levels of conservation within versus between strains?
Yeah, that's some ideas that we had, because this change is pretty dramatic. Just one single mutation, you got like dramatic killing. We did see other variations, but it seems to be difficult to find because the intergenic region, unless you see a phenotypic differences, it's hard to tell if that mutation matters or not because it doesn't affect the protein coding sequence. So this is like just lucky that we are lucky enough that to find one point mutation that is different between two different phenotypes, and that kind of gave us a hint that this is the mutation that we should focus on. I see. Yeah.
Any other questions? Oh. What? Oh, is there?
Oh, online questions? I think there's one more right there, but let me check. The mouse is always disappeared, so here. Nope.
Is it of interest to go into the actual coding region and find out how the SNPs that would affect the amino acid sequence or possibly create stops and indels, and how that would interact with the with this one one mutation outside that region?
Yeah, so there's definitely more mutation within the type 6 gene. To simplify the figure, I only put like one blocks right here, but they actually have a lot of like genes clustered together. It's a giant operon. And then this mutation is like throughout the operon. So it's kind of hard to tell which one is interacting with their mutation within the intergenic region. We also have a theory that the G to T is also affecting the bending of the DNA, like the minor groove and major groove. Yeah, I don't know if we can look into that and see if there's interaction. Right. Yeah.
If there are no other questions, I guess we could move on to the next speaker. I don't know who that is. Rozenn.
It's my pleasure to introduce Rozenn to this talk. If you don't know her, she's in the QVAS program, a PhD student. She's just a very creative scientist. She's also very caring. And she does both experimental and theoretical work all by herself. So it's a great pleasure to have her here.
Can you hear me okay? Yeah, I have to share my screen, right? It's already sharing? Oh, okay. I'll try. Mm-hmm. I see it here. You can't touch it. I'll try again. Oh, you saw it? I can't find it. We can't touch the screen. Still here. I think it's blocked.
It's so weird.
Yeah, I think it's blocked. This. Is it sharing? I think it's good. Okay. Okay, sorry about that. So the mic is on. Thank you for being here today, and thank you for introducing me, Pedro. So today I'm going to present the work I've done in the Ratcliff lab, which is going to talk about the emergence and the maintenance of stable coexistence in a Snowflake yeast long-term evolution experiment. And I did. Okay, is it better? No, it's not better. Yeah. Can you hear me better? A bit better? Yeah, I think it's good. Yeah, sorry about the technical issues. It's a bit loud now. No? Okay. So I'd like to start this talk by introducing a question that's of very big importance to biological development, and that's about multicellularity, and more precisely, it's about the transition from unicellularity to multicellularity. So how does, first I would like to give a simple definition of what a multicellular organism is. And as opposition to unicellular organism, a multicellular organism is just composed of multiple cells, of more than one cell. So how does one become many and in an organized way, a new one? So here I pictured the transition from one cell to a single cell. So how does this transition work, and how do we get this new unity that can function as its own? And in evolutionary biology, we call this a major transition in evolution, in individuality. And what is interesting about this kind of transition is that they have many consequences. And what I'm interested in is to study the consequences of this transition on diversity. So how does this evolve? Like, how does this transition, what consequences does it have on biodiversity? And what we know is that this simple transition to multicellular organisms actually had a great impact on diversity. So this is a phylogenetic tree of the fossil record from the Ediacaran. So you have the time on the x-axis. And what we can see really well is this very fast and dramatic increase in both the number of taxa and the diversity. So this simple transition to having more cells really increased the number of organisms and the types of organisms that we could have. Until the ecosystems that we have today. And of course, this was after millions of years of evolution. But I'm really interested in understanding how did we get there. And so in the lab, to understand this, we use experimental evolution on very simple multicellular organisms. And so I'm interested in understanding the processes underlying the ecological success of multicellular organisms. And our model study in the lab is a simple model of multicellularity that is called Snowflake yeast. So it's Saccharomyces cerevisiae. That's the species, but the multicellular form of the species in which the mother cell gives birth to a daughter cell, but they stay attached to each other. So it forms through clonal growth, and the way they reproduce is by simple fragmentation. So when a link between two cells is broken, we can have propagules, and this is the way they reproduce. And so in the lab, we make them go through a phase of growth, and then we transfer them to a new tube, and then we do what we call a size-based selection, where we're going to let the cluster settle to the bottom and only transfer the clusters that made it to the pellet to make it for the new round of growth. So that's the way that we grow them, and so we select for a large size. So we have a long-term evolution experiment going on in the lab that has 15 lines, that has 3 treatments. And this kind of experiments, they can go for as long as one's patients allow. And in our lab, this experiment is run by Rozenn Pineau, and he is very patient. So today we're celebrating nearly 1,200 days of evolution experiments. And the project that I'm going to show you today it just stems from a simple observation from one of his treatment lines. And we spotted the emergence of 2 different phenotypes, a small and a large phenotype. So from a simple monomorphic ancestor, as you can see from the confocal picture here, we saw the emergence of a small and a large phenotype. And so I looked at day 715 when I actually joined the lab. And so from this simple observation, we raised a few intriguing scientific questions, which was like, okay, we see these phenotypes, but are they really different? So both phenotypically and genetically. And how can we explain that they are coexisting? Like, what are the mechanisms promoting and maintaining this coexistence? And we used insight from both modeling and experimental tools to answer these questions. So first, I'm interested in characterizing and quantifying the divergence. So I isolated these 2 phenotypes, and I GFP tagged the small one. This is why it's green over there. And so you can see visually that there is a big size difference. So we decided to quantify it. So this is a size distribution of the ancestor, the small, and the large isolates. And we can see in yellow that the small is not different from the ancestor. However, the large isolate is about 4 times bigger than the small one. And the distributions are non-overlapping. So the size of the cluster is really different. But what about the cell inside the clusters? So we decided to characterize the cell shape inside the clusters. And to do this, we use aspect ratio, which is the length over the width. So the ratio of both can give you an idea of the cell shape. So the closer the aspect ratio is to 1, the most round is the cell. And so this is the result for the aspect ratio for the ancestor. So we see that the small clusters evolved rounder cell. As we can see on this picture, we can see they're really round. And the large cluster evolved nearly 2.5 times more elongated cells, as we can see here. So identified very clear phenotypic differences. And that's interesting, but we could think that they're being input in the media every day, and maybe they're just like they're reviving at every transfer. And so we were interested in knowing if these differences are heritable. And so to do this, we sequenced a few isolates of each population, and we found that within the smalls they were all sharing 16 mutations. Within the large, they were all sharing 9 mutations, but no mutations were shared between both. So if we make a phylogenetic tree of the small ones in yellow, the large ones in blue, we see that the last ancestor of the whole experiment is the last ancestor between both is the ancestor that started the experiment. So we found clear genetic differences. So as a summary of this first part characterizing the divergence, we see clear phenotypic and genetic differences, which suggest an early divergence from the ancestor and a long-term coexistence. So if we were to cartoon this, yeah. Can you say that again? Yeah, I mean, I think so. I think they started to diverge, but it's hard to say at which speed are they going towards each phenotype. But yeah, that's a good question. I will think about it. And so if we were to picture what we think is going on is that there was an early divergence, and this divergence has stayed for a very long time. So now, okay, we see this coexistence, but how can we explain it? What are the mechanisms promoting and maintaining this coexistence? So to test the stability of the coexistence, we competed the small and large clusters together. And so I started the competition with a different proportion of large and small strains. So if I start with 50% of large strains, there is 50% of small strain. And so I competed them over several days. And what we see is that regardless of the starting frequency, we always reach a stable equilibrium that is advantageous for small ones. So here we have 9%, about 9% of the large clusters, and so the rest is small clusters. Number of clusters, yeah. So, and this behavior is typical of negative frequency dependent selection. And what we further know is that the main limiting resource is oxygen. So we thought, okay, so they're competing maybe over this resource. So if we add more resource, if there is no competition anymore, what is going to happen? And so we did that. We added oxygen to the media. And what we see is that the small, they lose their advantage, and we disrupt the coexistence. So when the competition is released, we disrupt this equilibrium. And the small have an advantage when the resource is scarce. So then we decided to write a simple theoretical model to describe this experimental dynamics. And so this is the model. And what is interesting from the model is that not only can it recapitulate the experimental dynamics, but it can teach us more about the system. And so what we already see from this graph is that the long-term outcome predicted by the model is different from the experiment. So here we still have both strains living together, and here we see that we are slowly losing the small strains. And we think this is because in the experiment we can only spurge so much oxygen. It's not 100% all the time. But in the model, it's easy to set it to 100%. So we lose the coexistence in high oxygen in the model. So the small, they lose their advantage. So what this whole competition experiment suggests is that the small, they have an advantage when the resource is scarce. We also know that they have better access to the resource because they're smaller and their oxygen is diffusion limited, such that if they're smaller, the oxygen can just go deeper into the cell. So maybe the smalls can grow better. And the large clusters, because I showed you this settling selection at the beginning, so we select for large size. So we think that they will sink faster and survive better. So maybe there is a better survival for large. And so we test this hypothesis by using the model. And so this is the result from the model. And I would like first to focus your attention on the left of this graph, where I show the highest growth rate as a function of size. So you have the growth in black and as a function of size here. So what we see is that the smaller sizes, they will grow better. Now on the right of the graph, we can see the size-based survival as a function of size. So this is the gray, which simply tells us that larger sizes will survive better. And then I simply applied the experimental size distribution on this plot, which really shows that the size distribution kind of specialized into growing, and the large specialized into surviving. So there is this fundamental trade-off between growth and survival that drove niche partitioning that was simply based on size. So as a summary of this part, this fundamental trade-off between growth and survival results in the creation of two ecological niches, and this is simply allowed to the size difference. And so we see a functional specialization of small into growth and large into survival. So a few conclusions from this talk. We use theoretical modeling and experimental tools to show a stable coexistence from an initially monomorphic population. We describe niche partitioning based on size. And so the main message that we think we are learning is that the simple evolution of multicellularity without any adaptation can drive adaptive diversification and the expansion in multicellular ecological space. So I would like to thank my collaborators on this project, Ozan Bozdak, who did all of the experiments, David Demory, who helped me with the model, Eric Libby, who also helped me with the model, and my advisor, Will Ratcliff. And of course, I'd like to thank my lab. Thank you.
Are those small and large morphs, are they happening in the same environment, like the same test tube?
Yeah.
Okay. And then, so I understand the procedure by which you sort of like, separate them, you let them settle by size. And what happens if you disassociate the cells? Do they then form, like if you like have single cells that come from these morphs, these snowflakes?
So if you dissociate the cells from the cluster?
Yeah, then will they produce new clusters of their same type?
Yeah.
Okay.
Yeah, so they have a mutation that keeps the cell together, plus they have entrenched mutations with the evolution. So if you have a single cell from this cluster, it's just going to remake the same cluster.
So it'll recreate it. I mean, I'm wondering if there could be some epigenetic stuff that's separate from the genotype, right? That is sort of, if you never actually disrupt it and see if it recreates, like there is like genetic differences that makes this phenotype heritable. So it's not only, maybe there is some epigenetics, we didn't check that. Sure.
Okay. But cool.
It looked like there was a difference between the genetic divergence and the phenotypic divergence. It looked like the small ones were more genetically divergent from the ancestor, but more phenotypically similar, and vice versa. Is that right? And if so, what do you make of that?
Yeah, in terms of number of mutations, I agree. But then I think also it's like what does this mutation have in terms of phenotypic effect? And I didn't find any mutation that had big effects on the phenotype for the small ones. So that maybe explain why we see less divergence in terms of phenotype for the small ones.
Just a technical question. You got a divergence of 16 plus 9, 25 SNPs. How many cell divisions was that over? So 1,000 days, but how many cycles did you go through?
I have absolutely no idea. This is a very good question.
Okay. Like closer to 10,000 or one a day, 10 a day, 100 a day?
Generations per day. But how many cell divisions? Like, that's a lot.
100 cell divisions. So that's...
Yeah, that's a lot of cell divisions.
Okay. And my real question is, and first wanting to say that this is absolutely fascinating, are the number of cells in the large forms and in the small cells the same?
No. No,
okay.
The small ones, they have less, like, they have a very, like, they have less cells than the big ones. Yeah. And in terms of
very much less? Very much
less?
Yeah,
very much less. Yeah.
Okay, because that... Getting at the question of multicellular, you want to get at the question of multicellularity rather than how do you evolve being bigger. And the question would seem to me, what is the number of cells in each cluster is what you'd want to track. And intuitively, as you suggested, I just satisfied myself that surface to volume ratio of a tube is less than it is for a ball. So yes, the limitation there would be how do you get enough oxygen? But for multicellularity, can you grow a monster? The monster yeast that ate Atlanta. Do you?
I suggest you come visit us in the lab. Okay. So we have monsters, but they're anaerobic. So it's when they're not respiring oxygen.
if there are no more questions, then let's thank Rozenn again.
I'm happy to introduce our next speaker, Dr. Avery Davis-Bell from the Paaby lab. Avery started genetics research during her undergraduate training, and after getting her bachelor's in biology from Swarthmore College, she went on to do her PhD in human genetics with Steve McCarroll at Harvard Medical School. She's been in the Paaby lab since May of 2020. And she uses her computational genetics research focus to study allelic variation in C. elegans populations, specifically focusing on gene expression data.
Thank you. That was like such a good introduction to me and what I do. I appreciate it, and thanks so much for the opportunity to speak today. So we're interested in how and whether patterns of natural genetic variation show up as phenotypic variation, specifically in gene expression variation, which is itself a critical phenotype, and it's also a really important intermediate between genotype and many organismal level phenotypes. So what proportion of genetic changes end up changing gene expression? How does this happen? Where are they? Is this different across systems? Do changes that increase expression get canceled out by those that decrease it? And if so, how frequently? And one such characterization is whether changes occur in cis, like we heard about in the fantastic first talk, and so that's on the same molecule affecting its linked copy of the gene, or in trans, somewhere else, like in a transcription factor that might affect all copies of a gene. How often are gene expression differences driven by these different modes of regulation? Do they tend to come together at the same gene and reinforce each other or cancel each other out and compensate for each other? And this type of question has been studied in a lot of systems, but particularly good experiments to tease it out have not yet been done in C. elegans. And this is actually a uniquely good system for learning things from analyses of divergent gene expression. And that's because these hermaphroditic worms usually mate with themselves, with only occasional outcrossing to males and other populations. This means that they're isogenic or homozygous in nature. So they have these long lineages, these very independent populations with only occasional mixing or recombination between them. This means that we get these large sets of genes and variants that are co-inherited together as blocks, which is completely different than we see in most systems that are sexually reproducing, right? And so this is exciting because these sets of genes and blocks might interact, and so we might expect to see compensation between them happening in this system. But of course, whether that's happening depends on how much variation is actually in the system. Is there enough to compensate for? The elegans can really give us a window onto this as well, because the strains are very differently diverged from each other. So this is the tree of what the species look like, and it means we can choose natural wild-type strains that reflect the genomic diversity and have very different levels of divergence from the reference strain, which is the N2 strain shown here in gray. And so we're studying the 5 highlighted strains here. And so the experiment we've done with my colleague Han Ting, a research scientist who used to work in the lab, first did RNA sequencing of these 5 strains. She also crossed the 4 non-reference strains to the reference strain, generating F1s, right? So they had 1 copy of each of these genomes, and did RNA sequencing there. And what we get from these 2 experiments are first the kind of classical differential expression, difference in gene expression amounts between the strains, and also allele-specific expression. So when these genotypes are brought together, how much does each one show up? And we get numerical estimates of that. And actually optimizing the methods so that we could trust this was a huge part of this project that I don't have time to discuss at all today. So please do ask if you're at all interested in methods. But these allele-specific expression analyses are actually a uniquely well-powered way to detect cis regulatory changes and study cis versus trans evolution. And that's because any difference in expression between the alleles that we can detect must be governed by cis changes, because the trans environment is the global environment, and that's shared across the alleles. And additionally, this comparison is being made within individuals. So the stuff we worry about being noisy and varying between individuals, we don't have to worry about. So here I'm showing you for one of our F1 crosses, all of the genes for which we could detect whether there was allele-specific expression or not. So this is a volcano plot, and it has the proportion of reference alleles or allelic skew along the x-axis. So 50% in the middle is even, no allele-specific expression. Out toward the edges as well, we're seeing one more allele more than the other. The y-axis is the significance in a genome-wide adjusted way. And so each blue dot is a gene with allele-specific expression. Here's this picture for all 4 of our strains. And I want you to notice a couple of things. First is that there's a different number of points on each of these plots. And that's because only genes with SNPs in their coding regions that we can pick up by RNA sequencing can we detect which allele each read came from. So JU1088 in the bottom right-hand corner has fewer SNPs from N2. It's most closely related. Fewer of its genes have SNPs, so fewer where we can even make these levels of calls. The second thing is that these points are symmetrical around 50%. We're not seeing way more reference genome alleles than alternate genome alleles. And that's important because that's a type of bias that can affect these type of studies that we're not seeing. So what we want to do is we want to compare across these blue dots, these genes with cis regulatory divergence, our allele-specific expression genes. Are we seeing more or fewer of these across divergence from N2? We can't really do it by looking at these plots because there are a different number of points. So what I'm going to show you is looking across divergence on the x-axis, so the number of sites that differentiate these 2 copies within a genome, the reference genome from the alternate genome, for the proportion of informative genes that have cis-regulatory changes. So we expect the number of informative genes on the denominator here and the number of genes with detected allele-specific expression to scale with genomic divergence. But the proportion could stay steady, as in this top cartoon, which would mean we accumulate cis-regulatory changes at the same rate as genome changes. So each new change to the genome has an equal likelihood of changing gene expression. Or we could see this increase with more mutations, such that each new mutation has an increased chance of changing gene expression, maybe because more mutations are accumulating within genes at that point. So what do we actually see? Again, these are the real numbers of distance between the 2 genomes on the x-axis. This really looks like we're seeing the second scenario, whereas more genomic changes occur and accumulate. Each one is more likely to cause cis-regulatory changes and therefore be detected as a gene with allele-specific expression. But do each of these cis-regulatory differences therefore show up as differences in gene expression across strains? Are there also trans differences at these genes? Unlike differences between the alleles, differences between the parental strain, that more classical differential expression, can occur either in cis or in trans. So by comparing these to each other, we can tease out at individual genes and globally how much change is due to cis or trans regulatory variation, and do these tend to reinforce each other or compensate for each other. So what this looks like is, first, if we have a difference between the alleles in the F1 individual, we know that must be driven by a cis change because the trans environment is shared across those alleles. If we see that same difference in the parents, well, they've just taken that cis change right on up. That gene's change in gene expression is largely due to cis changes. On the other hand, if there's no difference between the F1 alleles, we know there's not cis regulatory divergence. And then if there is difference between the parental strain's expression, then that must have occurred in trans, which was shared between the alleles, but not between the parents. We could also see a case where there was a difference between the alleles, but no difference between the parents. So the difference between the alleles tells us there's a cis-regulatory change. The fact that we don't see it in the parents suggests that there would be something compensating for this, so a compensatory trans-regulatory difference. And we can actually look at this genome-wide all at once by plotting on a scatter plot on the x-axis the difference between the parental strains, and on the y-axis the difference between the alleles. So in this sort of space, along the y equals x-axis in kind of coral here, would be the cases where there's equivalent difference between strains and alleles. Those would be our genes with cis-regulatory changes dominant. Along the y equals 0 line in purple would be cases where there's difference between the parental alleles, but not the parental strains, but not between the alleles. And so those would be cases with trans-regulatory differences dominating. And then along the vertical spread of points, in yellow, would be cases where there was difference between the alleles, but not between the parental strains. So we know there's cis-regulatory difference, but there's something compensating for it. And I know that's a lot, so I'm going to keep the cartoon up. And here's an example of our actual data in the cross between the CB4856 strain and N2. So on the x-axis is the difference between those strains, and on the y-axis is the difference between those alleles when they're together in an F1. Each point here is a gene. The color is just based on whether the log 2-fold change is significantly different from 0. And what I want you to take away from this plot is we see all of these classes of genes that I just described to you. So there's a spread of points along y equals x, like that one in the top right-hand corner, that has kind of equal difference between the strains as between the alleles. You see the horizontal spread of points where there's differential expression between the strains, but we don't detect differences between the alleles. And this is only for genes where we could detect it if it were there. And then there's a vertical spread of points where there is a detectable difference between alleles, but not between strains. So there's a cis difference that is compensated for and isn't showing up as a difference across the strains. And of course, we're really interested in comparing this across strains, and just showing you a bunch of scatter plots, you can't really do that. So we want to ask, is there more compensation proportionally as you have more divergence across the strains? And so that's what I'm showing here. This is the proportion of genes with cis-regulatory changes that have compensatory trans changes. So it's that vertical spread of points divided by all of the ones with allele-specific expression we detected. And so what this shows you is like that the purple string, QX1211, it has more informative genes and more cis-regulatory changes. We knew that. But it also has a higher proportion of those changes that are compensated for. So we know there's cis differences, but then they're not showing up as differences across the strain. So as the cis regulation of gene expression diverges more, the compensation to correcting for this also increases. What might this mean? Well, recent analyses in C. elegans have demonstrated that, as in other species, strains are more different in DNA space than in expression space. This suggests that stabilizing selection acts to keep expression more stable, maybe at a more acceptable level. And so perhaps this is happening by increasing the proportion of cis changes that are compensated for by trans changes as more cis changes accrue. So instead of keeping down the number of cis-regulatory changes, those are happening, but there's some compensation going on. And of course, this makes us wonder, where and who are these genes with compensatory trans changes? And this and lots of other questions are our next direction. Like, are they physically linked? Are they in pathways together? Are they these co-inherited sets of genes? And can we figure that out? What are genome-wide expression patterns inherited, like additively, dominantly? How does this relate to the regulatory patterns I've shown? So lots of stuff to do. And I also want to briefly note that toward a goal of making research and results more accessible as living data sets for the community, we're also working to turn our results into Shiny apps. And so I have some of the data currently available and plan to keep doing that as I do new analyses. So yeah, look up your favorite gene. And I'd like to conclude by acknowledging all that went into this work, especially my co-lab members, Han Ting, who did the experiments, Annalisa for her constant guidance. Others at Tech have enabled the experiments and also the web backend, and external funding and resources are, of course, critical. I look forward to your questions, and I really appreciate your time and attention. Thanks.
This is a really technical question. When I'm looking, and it's nice that the volcano plots are there, do you have a means to, you said that, well, the left side and the right side look the same. Can you prove that mathematically? And can you prove, how do you, or if I look at the one in the lower right-hand corner and the one in the other upper left-hand corner, can you say that they have different shapes in any quantitative way?
This is a great question. and the issue is time. So on this side is a quantification for all of the genes, which ones are, so this may not answer your exact question, but it's, I think, closer than what I had time to show in the talk today. So which proportion are reference biased and which are alt biased? And so what you can see here is that, yes, the confidence intervals span 50%, which makes us happy. But what also makes me really happy is that there doesn't seem to be a trend with divergence. And I'd expect if I had a reference bias issue that the farther away the genome was from reference, the more that bias would show up, and I don't see that. And I have similar plots. I don't think I have a hidden slide for this, but also just restricting to genes that do have allele-specific expression. So where we're really trying to tease that out are those reference biased, and they aren't.
So you could apply a statistical test if you were willing to make certain assumptions.
Yes, yeah, and the statistical test of testing whether this is different from 50% is not significant. But yeah, I haven't done the exact shape test.
Very good. Thank you.
How do you distinguish between transcomplementation in the parental strain versus G by G interactions in the F1, or maybe like trans-stimulation in the F1? In other words, the assumption that they have to be cis changes could be violated given that they're placed in a completely different genetic background.
This is fantastic. This is a great question. I don't have the full answer to this, but it's really interesting. And some of what I want to do is at individual gene levels is actually these strains share haplotypes at parts of their genome and not at others. And so I want to actually narrow in on basically places that are shared to really test the assumption, is it cis? Because if it's cis, you'd expect if the haplotype is shared, one place you would see it even if it's in a different genetic background. So that's one way to get at that. But it's a really good point. You're going to make Thanks, Samiksha.
That's a great talk. I have a more broader question. So do you think this method can apply to other diploid but asexual reproduction organisms?
Oh, yeah. Yeah, absolutely. And this is actually, I didn't have time to get into a lot of background, but allele-specific expression, we've learned things from these analyses in lots of cool systems. And yeah, I think kind of anywhere you can get two genomes into a cell, you can learn things about it. And it's been done in humans, right? You can't compare to the parents that are isogenic, sadly, because we can't, homozygous people and then have them mate. But you can look for kind of QTL type stuff in naturally heterozygous populations.
Any other questions? I think we can move on to the next. Thank you.